The top protein sequence consists of the amino acids methionine, tryptophan, phenylalanine, threonine, lysine, and glycine. The bottom protein is the mutant one. It consists of methionine and tryptophan, and then it differs when it has isoleucine and histidine. Now we want to determine if this mutation was caused by an insertion or a deletion in the mRNA. So we're going to write down all the possible codons that could have coded for the amino acids. For methionine, it's AUG, tryptophan, UGG, phenylalanine, UUU or UUC, threonine, ACU, C, A, or G, lysine, AAA or AAG, and glycine, GGU, C, A, or G. Now to determine the mutant codons, we have AUG for methionine, UGG for tryptophan, AUU, AUC, or AUA for isoleucine, and CAU or CAC for histidine. Next, we use the technique that we learned in problem session. We match up each base with the corresponding one from the normal mRNA to the mutant mRNA. Once we run into something that doesn't match up, we realize this is where our mutation occurred. In this case, A was inserted into the mutant mRNA. We continue matching everything up until we have a complete coding sequence. For the mRNA, this coding sequence is 5' AUG, UGG, AUU, CAC, 3'. For the normal mRNA that occurred before the mutation, we have 5' AUG, UGG, UUC, AC, and then for the next ones, it could be anything because we don't know. So I'm just going to use the top ones because that's easier. ACU, AAA, GGU, 3'. At the end of these mRNA coding sequences, I could have added a stop codon to signify that protein synthesis has stopped. However, these are just portions of the actual protein, and I don't know how long they go on for, so I decided not to. In summation, this mutation was caused by an insertion of an adenine base in the mRNA. Base pair insertions and deletions cause frameshift mutations. This is a change in the reading frame for all of the codons following this mutation. In this example, the insertion of the adenine caused the reading frame to shift, so instead of phenylalanine, we got isoleucine. Then after that, everything was changed, so instead of threonine, we got histidine, and that would continue to change for the rest of the protein, unless of course it ran into a stop codon after this.